All right, thank you, Craig, for the introduction. I will be speaking about reprogramming human stem cells to study cardiomyopathies. I'm James Ellis, and I'm at the Hospital for Sick Children. This slide here shows the concept of what reprogramming is and how it can be used for disease modeling, for drug discovery, and I'll describe some new work that we've been doing with Seema Mittal on evaluating functional genomics using these patient and control iPS cells. So what's shown here is the process of reprogramming, where blood cells or skin cells can be taken from a patient or a control individual. Using 4 genes, they can be reprogrammed so that they start to grow like embryonic stem cells. They're called induced pluripotent stem cells, or iPS cells. Then these cells can be differentiated into any cell type of interest, and in particular here, we're interested in cardiomyocytes. So now we have cardiomyocytes derived from a patient or from a control individual that can then be used in a dish for disease modeling. So in the dish, the cardiomyocytes would start to beat, and you can measure the beat rate, the beat amplitude, and various other measurements that I'll describe later. Once you've found a cellular phenotype, as it's called, you can then use that for drug screening to see, will a drug correct the phenotype that you've identified. And in my talk, I'll describe some disease modeling in the end of this talk in the second part. What we have started to do now is functional genomics. And what functional genomics is that you can use these cardiomyocytes to examine the effect of genetic variants that don't knock out the protein, the coding region of a gene, but actually impact how much of the protein it makes or how much of the RNA it makes. This is a slide describing the current state of cardiomyopathy. It was provided by Dr. Seema Mittal. And the situation is that it's very polygenic. There's over 100 genes, and they're shown here with the different types of cardiomyopathies that can be mutated and lead to these different types of cardiomyopathy. So for instance, here's myosin heavy chain 7 and myosin basic protein C3, which I will describe later. Now, when genetic screening is done, unfortunately, only 30% of individuals are found to have these coding variants in these genes, and all the rest of the individuals are gene elusive. It's not clear what the genetic mutation could be. And so it's possible that these gene elusive variants may be in the promoters, which are the control elements of a gene, of these known CMP genes, and affect their expression. So that even though the protein that made is normal, there's less or more of it, and that can impact and cause the disease. So this is a summary of work doing whole genome sequencing by Seema Mittal's group. And what it shows is that there are novel regulatory variants in these CMP genes that actually cause 20% of pediatric cardiomyopathies. And these have never been seen before. So now you're going from only 30% of people having an identified variant associated with the disease up to 50%. In black are the ones that have previously known for having coding mutations. And over here is myosin heavy chain 7 and myosin basic C3 that I'll describe later. But in red, all of these are new regulatory variants. And we're going to focus particularly on this dystroglycan subset here of DTNA and fucotin. Over here is an analysis of the genetic burden by comparing these variants to control databases. And they show that these variants are much more prevalent in the patient samples. And again, the ones that we're interested in particular here are fucotin and DTNA. This is an example of 4 of those genes that have these predicted regulatory variants that we expect would affect something called the dystroglycan glycosylation pathway. These are the 4 genes: ddNA, FKRP, FKTN, which is fucotin, and large 1. If you look at all of these proteins in this pathway here, this dystroglycan pathway, you can see the red ones, the ones circled in red, large 1, FKRP, and fucotin, are all involved in adding these glycosylation subunits to dystroglycan. And DTNA over here, or dystrogliflozin, is also associated with this phenotype. So they clustered in one particular phenotype, and these patients, there's no previous suspicion that they have anything wrong with this particular pathway. So how do we test whether those regulatory variants that would affect gene expression actually do affect gene expression? And for that, we can use stem cell-derived human cardiomyocytes. So here we've got healthy control, a cell line called PGP17 that we made from a healthy individual and reported in this paper last year. They can be reprogrammed by the addition of these 4 genes to make induced pluripotent stem cells. And then using a kit provided by a Canadian supplier called Stem Cell Technologies, we can make immature cardiomyocytes that are beating, that then mature somewhat, and we can test by comparing these regulatory variants to their normal variants, whether there is an effect on gene expression. In the later part of the talk, I'll describe how to make the patient iPS cell lines and take them all the way through to doing that cellular phenotyping and drug screening. So this is what's called a luciferase assay, and luciferase is a gene that when you make it, it emits light. And so what you can take is the wild type or the reference sequence to see how much of this light producing protein gets made and compare it with the regulatory variant. You can then introduce these into those human cardiomyocytes that we've made using the process I described earlier, detect the light on this device here, and then look to see, is there an impact of the regulatory variants? These are controls that just show that if we have a fully functional promoter element, that it works well. If we have no promoter element, it doesn't work. This is in gray is the reference normal sequence, which shows how much of the luciferase you're making. And then the comparison with the regulatory variant, which shows that the expression level is much reduced. So that's the first evidence that these novel regulatory variants actually affect gene expression. Another thing that you can do is something called massively parallel reporter assays, or MPRA, to measure the effect of regulatory variants compared to the reference sequence, again in the stem cell derived cardiomyocytes, but doing many genes all at once. And the way this process works is you take the regulatory variant and its normal sequence for comparison. You introduce these small DNA sequences into this particular construct here in this location. And what you're looking to see is, does the alternative allele, the regulatory variant, reduce expression of this? So these constructs get transfected or introduced into the beating cardiomyocytes. And then if you sequence the RNA that's made by these, you can see how much of the reference transcripts do you get in comparison to the, the variant, and that will give you an indication that that particular variant affects gene expression. When you look at 52 variants that Seema's group found in the genomics, you can see that 29 of them have either more or less expression of those sequences. And then the final stage that you can do is look back at actual patient material. So an individual who has a mutation in the fukutin promoter, this is the DNA sequence that surrounds the variant. It is normally bound by a regulatory transcription factor. When it's mutated in the patient to an A, that factor would no longer predict it to bind. So if we look at RNA sequencing and RT-PCR, which is a way of looking at the RNA levels in this particular patient compared to all the other patients, we can see that the fukutin RNA is reduced, and that's confirmed using this qRT-PCR. If we look at protein levels, this is protein, again, from a biobank sample of the individual compared to other individuals, and you can see that the fukutin protein is also reduced, and that's quantified here. And the last thing we've been able to do is to reprogram cells from this individual to make those iPS cells, differentiate them into cardiomyocytes, and then look to see how much of the fukutin RNA is made in these cardiomyocytes. And again, you can see it's reduced. So all of this adds up to this particular point mutation here seems to reduce expression of fukutin. And what we're doing now is we're correcting fukutin, the variant in this fukutin iPS cell line to see, does that correct expression levels of fukutin, and can we then use it for disease modeling? So let's move to the disease modeling. We've been working for a couple of years with Seema Mittal's lab and a few other groups, including Craig Simmons, to use precision therapeutics for cardiomyopathies. And in particular, we're interested in testing myosin inhibitors in hypertrophic cardiomyopathy, or HCM. And myosin, those two genes, myosin heavy chain 7 and myosin basic protein C3 variants, account for the majority of the genetic causes of cardiomyopathies. And so we wanted to test new therapies in the stem cell derived models from patients with these variants to determine if there is a variant-specific response. And we're in particular interested in this mavacamten, or MIC-461. It's used in clinical trials in adults with HCM, but is not being studied in children. And what mavacamten, or MIC-461, does is it's the myosin ATPase inhibitor, which would prevent or reduce activity of the ATP binding here to the myosin heavy chain 7. When we went ahead and made iPS cells, we can differentiate them into the cardiomyocytes. This is staining for some cardiomyocyte-specific proteins over here, where you can visualize these cells and measure the myocyte area. Here are 4 different individuals. 2 have mutations in myosin heavy chain 7, 2 in the myosin basic protein C3. And if you look at the myocyte size, you can see in the patients it's much larger. Once the patient's variant has been corrected through gene editing, it now goes back down to the normal level. That's the case in all 4 of these individuals. You can also look at the sarcomere alignment in these cells, and you can see that in the patients, the alignment is very poor. After it's been gene corrected, it has improved to normal, with the exception of this individual here, because we only corrected this mutation, and it actually has a second mutation present. And finally, we can look at the function of these patient stem cell-derived cardiomyocytes. And what this slide shows is that they have a higher beat amplitude, a higher calcium flux, and they respond to the myosin-targeted drug mavacamten, the MIC-461. So these are healthy control cells, and on an Excelligence device, you get these wave forms which show the beat amplitude and some extracellular voltage waveforms. When you look at the beat amplitudes, this is the control cells here. You can see that they're fairly steady, and all of the patient material has this increased beat amplitude. If you look at how calcium is handled, you can see that the calcium this calcium flux is higher in the patient, and it gets reduced down to normal. And then if we do this assay over here with the drugs, what we see, we're just looking at a single patient here, is treating with just a standard vehicle compound has no effect. If you treat with 461, you can see that you reduce the beat amplitude back down to a more normal range. And standard drugs like metoprolol and verapamil have no effect. So that's an example of how one does the disease modeling and the drug screening. So overall, in conclusion, what I've shown you is that functional genomics in stem cell-derived cardiomyocytes demonstrates the effect of regulatory variants on expression of these CMP genes. And what's, I think, quite impressive about these findings is they identify druggable dystroglycan or other pathways that were previously gene elusive. In those individuals, you didn't know that those pathways were mutated because they didn't have coding variations within the gene. It's regulatory variations upstream of the gene that control it. I also showed you that HCM patient and corrected iPS cell lines demonstrate the expected cardiomyocyte cell phenotypes and respond to the myosin-targeted drugs. And in our next step, what we're trying to do is we've got the Fukutin iPS cell lines. We want to look at their cardiac phenotype to see, are they behaving abnormally? And then if they are, we can test the effects of dystroglycan glycosylation targeted drugs on those cell types. So finally, for acknowledgements, the heavy lifting of the work that I've described here has really been done by Seema Mittal's lab. The genomics was done by Robert Abdo and Oye in collaboration with Steve Scherer's lab. Abdo did most of the gene expression studies. Carolyn and Neha did the disease modeling work. And my lab is more of a supporting lab in that Guo Liang in particular has produced the cardiomyocytes that have been used for all of these studies. And Philip Mass's group was instrumental and crucial in doing those MPRA assays. And thank you very much for your time.